and you're familiar with the proton gradient again in that picture, the big H in the intermembrane space, while the little h in the matrix, right? And now the third part of oxidative phosphorylation involves an ATP synthesizing enzyme. What is that ATP synthesizing enzyme called? ATP synthase. That's the ATP synthase. And where is it? It's also embedded in the membrane, isn't it? It is the proton gate, isn't it? When the protons go through the gate, it spins the gate, doesn't it? Okay? So that's also embedded in the inner mitochondrial membrane specifically. In this case, we're just going to say in a membrane. And this ATP synthesizing enzyme embedded in the membrane is going to use the proton gradient to do what? To make ATP. Bingo. So that's oxidative phosphorylation. Can you see why it's called indirect ATP production? It involves all these intermediate steps, doesn't it? While substrate level is simple and just a straight transfer of high energy phosphate, here we first get high energy electrons. Then the high energy electrons make what? A proton gradient. Then the proton gradient makes ATP. Okay. So that's the picture we have. And again, I would refer you to, I believe it's 24.8. All of that has oxidative phosphorylation. And so we have three parts now of oxidative phosphorylation. Which part is chemiosmosis? Just the third one. That's it, isn't it? Very good. So here's chemiosmosis. So that's how you distinguish between chemiosmosis, this step, one part of the three parts of oxidative phosphorylation. Good. All right.
They're high energy electrons because of the phosphate,
the electrons happen to be high energy relative to the electrons at other states. It just so happens that the electrons in fats and carbohydrates are high energy and they're usable for that purpose. Yeah. So now we can restate this idea of chemiosmosis, which is, if you think about it now, let's see if you can get this. Let's see what we're going to do. We have a cell now. And it has a low concentration of glucose inside. Okay? And, excuse me. It has a high concentration of glucose inside. It wants glucose. And we have a low concentration of glucose outside. But this cell really has a sweet tooth. And it wants even more glucose. So if we pump glucose from low to high, what is that process called?
Low to high would be active transport.
That's active transport. This is active transport, isn't it? All right. Now, let's go, and I'm going to just draw a really crude, but a big one, mitochondria, right? So that I can create a really big H plus here and a little H plus here. And in this case, the big H plus is going to go the high, this is high concentration, this is low concentration, that proton is going to pass through what enzyme embedded in this membrane, this inner mitochondrial membrane? ATP synthase. And we go from high now to low and make ATP, instead of using ATP to go from low to high. What are we going to call this?
Facilitated diffusion through ATP.
Not passive transport, not facilitated. This is the reverse of active transport. With active transport, we used ATP to go from low to high, right? It takes ATP to do that. Here, we're going from high to low, but we're making ATP. Folks, you know that most of the time when you go from high to low, you don't get to make ATP, right?
Right.
That's happening all the time in your cells. That's just called simple or facilitated diffusion, isn't it? You don't get any energy. Here's a case, a very special case, where we go from high concentration to low concentration and we make ATP. Reverse of active transport. That's what it is. It's really
So
the
that's
reverse
actually
of
what it's called, reverse
I mean,
of active transport?
I'm calling it that.
Oh, okay.
I'm calling it that.
Jerry.
No, yeah, but I'm calling it that to try to get you to think about what it is, what's happening. That's all it is. It's nothing more than the reverse of active transport. It's not magic.
Well, isn't
So
that
is
the
it
part
still
that's
active
chemiosmosis?
transport?
It's not active transport.
I don't understand.
But
It's
isn't
not
that-
active transport, because active transport would go from low H plus to big H plus and would require ATP. We're going from high to low and we're making ATP in the process.
So isn't that the chemiosmosis?
It is the chemiosmosis.
Yeah.
So chemiosmosis is the reverse of active transport.
Oh,
That's what I'm trying to say.
Yeah,
That's what I'm trying to
got
say.
it.
It's chemiosmosis is the reverse of active transport.
Well, it's osmosis in chemical.
That's right. Now, in that homework on oxidative phosphorylation, oxidative phosphorylation, stop and think about it that I gave you. By the way, the answers are already posted on the webpage. If you've tried to answer them, you've studied, tried to answer them, then it's time to go look, okay? But try to answer them on your own first. I talk about a chemical that was used as a diet drug called dinitrophenol, okay? Don't use this. It's not legal anyway. Dinitrophenol will cause you to lose weight. All right, let's see what it did. People were using it, they had no idea what it was doing. What dinitrophenol does is it puts a bunch of holes in the inner mitochondrial membrane. It becomes leaky. So we eat the banana split, right? We get ATP, we get lots of reduced NAD and FAD. We get a proton gradient, and the protons can move back into the matrix through ATP synthase, or they can move back in through what? These leaky holes. When they move through ATP synthase, we get ATP. We get energy. But when they move back through the leaky holes, what do we get? We get nothing. So you had calories that you took in, but no energy use out of it. No ATP that's usable. So that's just a point
So
there.
how would it help
Lose your
people
weight,
lose
yeah.
weight?
I
What
didn't
would
understand
happen
that.
to that?
Well, what would happen to that is you would have to start digesting your fatty reserves because you're going to about 40% efficiency of this process. Instead of a high level of efficiency, you're going to a low efficiency, and you don't get much in the way of ATP from the food that you eat. So you have to supplement the food you eat with your fatty reserves. But this is a dangerous drug. I really mean to imply that. I use this example because I think it's kind of cool what it does. It just punches holes in, and then we can see how that can harm. What specific process is harmed? Chemiosmosis is harmed by dinitrophenol, isn't it?
But once that happens, how is it going to repair itself?
It repairs itself in the same way that all